Hello everybody. I am Atsushi Kawaguchi from the Biology Group in Institute of Medicine, University of Tsukuba. Today I'd like to talk about the molecular biology of emerging biological questions. First of all, perhaps everybody already may know, but I'd like to explain on the basis of the viruses. This slide indicates the general principles of the viruses. First, the virus consists of the viral genome and the outer shell. This outer shell, some of the viruses have the protein capsid, but others have the lipid membrane originated from the cellular membrane. And you know the size of the virus is smaller than our 1 million cells, so the size is around 100 nanometers, so the 1,000 times smaller than that of the 1 million cells. So the size of the genome is also relatively small. Thus, the number of genes is also very limited. In the case of influenza, viral genome encodes only 12 viral proteins. In the case of SARS-CoV-2, the virus has 26 proteins. So only 12 proteins not enough to replicate itself. The virus replication requires cellular protein in host cells. Here is the empirical formula that this carbon 6,062, carbon 52, but anyway, it counts the number of humans or other cells. But in the case of the virus, we can write as like this. And then this one is recorded by Carl. And then he said that outside the host cell virus is as a pinpoint ball. It is a chemical that has been homogeneity and crystallized with its chemical bodies largely determined, and its three-dimensional structure solved, just like common chemicals has been synthesized in the test tube. However, once polyvirus chemical has entered the cell, it has a plan for survival. So this means that virus is similar to the seed of the plant. When it encounters an environment that allows it to sprout viruses, the virus will take shape of the living organism. So the question is, what is the emerging viruses? We mainly focus on emerging infectious diseases, especially related to zoonosis. On the Earth, there are three types of animals: human, wild animals, and livestock. Each animal group has specific infectious diseases. Some of them cycle between each group. SARS-CoV, Ebola, influenza viruses. Suddenly, these viruses spill over all these lines and transmit to humans as emerging viruses. what happens in just a priest viral proteins need to ability to use derived into different hosts. I mean from the human. And next, in general, virus infections are restricted by immune response. Thus, the viruses need to escape immune responses. So, this is the case of influenza. The natural host of the natural viruses is thought to be that, and there are more than 100 subtypes in that, but only three of them become seasonal influenza in humans. When avian influenza ability to infect humans, it is necessary to acquire mutations in intermediate host chickens and pigs, and furthermore, avian influenza finally kills humans. There are a lot of already reported adaptive mutation. The detailed how the adapt to humans this remains unclear. So most important questions are how the host length is restricted, and the viral pathogenesis is in humans. Then to understand these, first we need to understand how viruses using their host factors. So the important concept of this is that a little bit explanation about viruses has a cell factor and machine is replication. And then to fight with this virus infection, humans activate antiviral response. Thus, virus need to escape this antiviral response. So we need to understand possibly negative virus replication. This slide indicates the transcription mechanisms of influenza. Influenza virus has system of the PD1, PPA, 3 and the nucleoprotein carbon the virus genome into a ribonucleotide BRP. First, the viral polymerase cellular messenger RNA is a cap structure. Then the PA subunit cleaves this cellular messenger RNA. This RNA is recruited by the genome for transcription as primary messenger RNA synthesis. So far, we try to reconcile this capping. This process is called transcription mechanism. And so far we try to recap snatching and transcription mechanism in vitro biochemically. But here is cap snatching activity of the cellular polymerase obtained from seasonal influenza, avian influenza, but humans. This one is avian influenza, but this one is very close to influenza. then the top on top you can find this is before caps Latin reaction. Then by adding this cap RNA to the short RNA synthesis. Then on the light panel you can find quantitative results. Seasonal influenza is the slug activity. However, in the case of the influenza, we shows the biopolymer activity compared to the human influenza. So we can say that the adaptation is quite great. It's activity. So to understand this, now we start to structure analyze to understand how avian biopolymerase increase its biopolymerase activity the adaptations to humans. So to address this, now we apply analysis using this is the one of the electron microscopy, and then this is the one of the example. Then you can find the biopolymer structure on bioelectron microscopy. Then we try to reconstruct the structure for the RNA synthesis as like this. Anyway, I like to SARS-CoV-2. This slide indicates the symptom in this slide. A virus and SARS-CoV-2 respectively. In the case of influenza, this virus infects to lower respiratory tract and induces massive pain. And the case of the severe virus, sterile myocarditis. These are totally induced cytokines, overexpression of cytokines. And then in contrast, in the case of SARS-CoV-2, this virus is thought to be very serious infectious diseases with the influenza A virus. So SARS-CoV-2 also infects cracks and stimulates cytokines. This results in headache, muscle pain, and so on. But in the case of SARS-CoV-2, this virus can transmit the blood is called viremia, and then finally to the other organs, and then the result of the multi-organ failure. So now the question is, it is so to sort that influenza A virus and SARS-CoV-2 are very similar respiratory diseases. However, SARS-CoV-2 can transmit to the bloodstream. So to understand this, we did some cell biology experiments. So this is a wide field we choose infected cells. SARS-CoV-2 infected cells were infused into giant like cells, and somehow this giant cell expand unique structures in our heads. And as shown in this enlarged panel, some large structures were also observed. Then we found that these structures is one of the hatching related structure called invadopodia. But anyway, I'd like to skip the detail. Anyway, what happened this, we detected viral proteins, LSP8. This is one of the viral proteins, and this is marker for viral replication. They are shown is our hands, and large color. Viral polymerase accumulates the leading edge of fluid structures, and then we found that double-strand RNA in this structure. So suggesting that virus infection induces the membrane extension. And then in the fluid structure, somehow the polymerase leads edge of this neural-like structure. And then this is one of the protocol SARS-CoV-2 induced by anemia. I'm sorry, I omitted the detailed results, but SARS-CoV-2 type 2 pneumocytes, and then here is endothelial cells. In general, the basement to separate pneumocyte and epithelial cells. However, infection induces invade polio finally results in the degradation of the basement, and then this structure, the replication sector, and then from here, possibly viral particle is released to the bloodstream. So next, let's go to the next topic. Related to the immune response. As you may know, the virus infects the epithelial cells, this results in the epithelial cell and secretion of cytokines, islet beta. The neutrophils and the macrophages are migrated that engulf the infectious agent and infected cells. But if the macrophages are infected with different viruses, these produce much amount of cytokine, then this finally induces the cytokine very severe pneumonia. So we can say this epithelial inflammatory response is one of the important antiviral response of the past defense against virus infection. But the detailed kind how epithelial cells by infection, this remains unclear. All hypotheses to understand this response in epithelial cells. Then we actually try to identify sensor molecules of the vaccine to understand how epithelial cells decolonize those cells. And finally we identified mixing NXA. This protein is the type 1 internal inducing gene, and this is one of the dynamic large GDP. It is known that one of the determinants so far. However, its action through inflammation remains unknown. This is a summary slide activating inflammatory response. Infected epithelial type 1 type L then the interval weights are special and that's why the NXA next recognize the bias impression. And then after that NXA the iron beta production and then iron beta neutralize neutrophil to remove this infected cells. Anyway, this is in vivo results related to MXA program. Now, this is Japanese character, but here is late. And then, in the case of non-TG mice, upon virus infection, all mice cured by influenza virus infection. However, the NXA gene to the mice, these mice shows the resistance against virus infection. Then, as I told you, the nature of influenza is to be hence the avian influenza receives active mutation, then finally replication activity in humans. Among such adaptive mutation in NP gene also escape mutation against NXA. So this slide indicates the special level of the iron virus infection. These lines are H5N1 and H5N7 avian influenza, and then blue, purple, and black lines are human influenza. As shown in this green and red lines, avian type influenza quickly induces island beta suggesting that LSA easily recall these virus infection. However, in the case of human case, the secretion of iron beta was delayed compared to avian influenza. So this is due to the escape mutation in NP against NXA so suggesting that A is thought to be one of the determinants of the avian influenza. So this is a proposal model. Avian influenza humans and then to fully adapt humans, the virus needs to receive adaptive mutations to escape from to become influenza. So anyway, thank you very much for your kind attention. These